Yeah, I'm going to talk today about some just drug studies that we did or have been doing in mouse models of KCNT1 epilepsy. Um, this is a chart showing that all the recurrent variants, or at least a good chunk of the recurrent variants that have been found. This is from a nice review a couple years ago in Brain, which I would highly recommend to get a nice overview of the clinical phenotypes of KCNT1. But I have red boxes around two of them here. One of them is the Y796H variant, which, as you can see in at least in this publication, has been found in 5 patients, and exclusively, at least in the patients so far, has been found to be associated with sleep-related hypermotor epilepsy. And then below that, then is the R428Q variant, which is been found in more patients and is primarily associated with the EIM-FS phenotype. So we've created mouse models of both of these variants, and one of them was published a few years ago. This was the 796H human, which is 777H in mouse. We collaborated with few different groups, primarily at Columbia University at the time, especially Wayne Frankel, whose lab did these EEGs, which I'm showing here. But you can see in panel A there is showing the nucleotide change that then causes the amino acid change. B is a diagram of one of the subunits of the protein and where the Y77H variant is. And then what we found in this model was that we found 2 types of seizures, ones which were generalized tonic-clonic seizures lasting about 30 seconds, and then another type which we call tonic seizures, which are shorter, clearly electrographic, and they are accompanied by jerks and twitches and the body movements of the mouse. And then if you look at C, that shows that in the homozygous mice, at least, these seizures were variable from mouse to mouse, but fairly frequent, and they, in keeping with the disease, were more likely to occur in the lights on period for these mice. And subsequently, we showed that they were they primarily occurred during non-REM sleep. So that's the YH variant. In the meantime, as I mentioned, we made another mouse model, again in collaboration with Wayne Frankel's lab, the 428Q in humans, which is 409Q in the mice. And we've characterized the seizure phenotypes of these as well. And it's pretty, even though it's a different variant and in humans causes a different disease, I would say this mouse in some ways might have stronger phenotypes, especially in the heterozygous, but I'm just going to show the homozygous today. And in the homozygous state, they have pretty similar phenotype to the YH mice. They have these generalized tonic-clonic seizures, as well as the tonic seizures. And there's kind of some early data showing the seizure frequency in this line, which you can see. This is also homozygous mice. Looks pretty similar to the YH line. But at the right there, you see that plot is seizures over time. So on the x-axis is 24-hour time, and on the y is how many seizures are occurring at each hour there. And you can see there's not so much of a circadian rhythm to the seizures as there was in the other model. Okay. So we are interested in, knowing if we could treat these animals with a small molecule to stop the seizures. We collaborated with Actio, which is in San Diego, which has done a lot of work, in drug discovery, and generated a series of compounds that block the channel with high potency and high selectivity. And so as you can see here, this is the general workflow of what we did to test whether these compounds would reduce seizure frequency in both of these models. So on the left, we've tested 2 compounds, and so on the left, there's a compound, for these purposes called ABS 569. We treated this compound, both lines, with 10 milligrams per kilogram dose, and characterized in this plot, you're just seeing the kind of combined frequency of seizures for both those generalized tonic-clonic seizures and the tonic seizures. We did a vehicle treatment period, we did a drug treatment, and then a washout period. So on the left is the data from the RQ model. So you can see during the vehicle, again, the mice are variable from mouse to mouse, but drug treatment definitely reduces the seizure burden in the RQ model. On the right there is in the YH model. This is the same compounds, same dose. it also reduces the seizure burden. And then on the right, there's a different compound that has some, different pharmacokinetics, and we've tested that one so far in the RQ model, and it also gives good seizure reduction in this. And so we're doing some more work with putting them on neurons to see how it changes, using these as a tool to see how it changes neural firing. Getting at the kind of the questions that Roy just showed in the previous talk very nicely. So we think this is encouraging data, and yeah, I'll thank everyone that worked on it, especially in my lab. Jang did all these EEGs, did a lot of injections for these mice. It's really a lot of work. Other people in the lab, and then of course Actio, the people at Actio that we've worked with, they've done a lot of in-house work to generate and characterize these compounds. So yeah, be happy to answer any questions.
Can I ask, do you see any AEs with these compounds when you inject them into wild type mice?
Have we seen any adverse effect? That's
Yeah.
what A.
Yeah.
No, not obvious ones. Yeah.
They don't fall off the rotorod or things like that?
We haven't tested them in any like specific behavioral test on this with this compound, but I mean we have video. We do video EEG, so we have 5 days worth of video that we go through when they're treated, and yeah, there's nothing obvious. Like they're not passed out or
Right.
running around the cage like crazy. So.
Good.
Hey, Matt, that's really beautiful work. Quick question about the washout of the first drug you showed. Maybe the prior slide may illustrate my question.
Yeah, I think I know.
Almost looks like there's rebound.
Yeah.
Is that statistically different from vehicle, or is it still preliminary?
I think that so this is a long-acting compound, so there's 3 weeks of rest in between that drug treatment and the washout. So I think what you're seeing is those mice they're aging, and their seizure burden is kind of getting worse as they get older. That's what I think, but I think that's why so on the right there, that compound is a much shorter acting compound, so it's only a week washout. I think that's what it is, but it probably would be higher, at least in that RQ. Probably is significantly greater than the vehicle.
Thanks,
Matt. This is really fantastic work. This is David again.
Hi.
I had 2 questions. One is whether your mice, what the behavioral phenotype or cognitive phenotype is in the mice, and if they're different between the 2 models, and if you see any behavioral changes after seizure suppression.
So with the YH, we characterized that in the first paper at the Columbia Behavioral Core. They had, at least at that age, I think they change over time. If you did these behavioral tests at different times during their development or lifespan, you'd probably get very different results. But at that time point, they had hyperactivity in the open field. They had some deficits in fear learning, and then pretty consistent change. The only one that we've measured also in the RQ is the nest building. They don't build nest as well, which is common to a lot of these models. We haven't done a detailed characterization behaviorally on the RQ. So if anyone wants to collaborate on that, let me know. And we haven't done much with the drug in terms of looking at more subtle behavioral phenotypes. So just seizures really at this point.
Gotcha. It'd be cool to just look at nesting as well. Just after treatment, you'd think that would improve. My other question is whether you thought about trying this compound in like the SCN8A or SCN2A mouse model, just to see if that previous work that had been done that showed improved mortality in the mouse model, if that would work with these other channelopathies as well.
Yeah, we've definitely thought about that. Would like to do that. I think one thing we're interested, because we've looked at inhibitory neurons in the past, and especially in the SCN1A, because of the potential mechanistic overlap that we see, is would this also have a benefit in that model, and also is there do they both basically help interneurons fire better? Do they improve their firing frequency? Because we do see a deficit in interneuron firing frequency in in both of these mouse models, neurons, and of course they also do an SCN1A. Yeah, and SCN8A may be a little bit different, but yeah, it'd be interesting to try that too based on the ASO results.